Okay. Good afternoon, everybody, and welcome to Wall Street Reporter's next Super Stock live stream for today, March 19th, 2020. I'm very proud to be bringing you two great companies, which many of you have been following since they first presented at our live stream conferences back in August. That's CytoDyn and Nextech. Both of these stocks have skyrocketed over 300%. Since they first presented their story to our audience. And of course, the market has had some turbulence. Both of these stocks have gotten knocked around a little bit. But the key point is this. The CEOs of both CytoDyn and Nextech, they've been here, they've been in front of investors, engaging with investors and shareholders from the beginning when the stocks were cheap, when the stocks moved up, when the stocks came down, and here they are again, which is a key point because a lot of companies, as a lot of microcap investors know, a lot of companies they hide when the markets turn. So really, this is the mark of true leadership that the CEOs are here with you today in these crazy times. So for those of you who are new to Wall Street Reporter's live stream, the next Super Stock live stream, the premise is simple. We're looking for stocks which have 10x upside potential. So these are companies which have they're going after multi-billion-dollar market opportunities. The companies are at key inflection points, and they have multiple catalysts in place which can drive stock price appreciation in the months ahead. And most importantly, they have a CEO that gets it when it comes to maximizing shareholder value and also transparency, as you're going to find out today. So please watch the presentations, ask your questions. It's your questions that really make these events special. And then decide for yourself if this is going to be the next super stock. So without further ado, please let me introduce you to Dr. Nader Pourhassan. He's the CEO of CytoDyn. Nader, take it away.
Thank you, Jack, and thank you everyone for being on the call today. With me today, I have Dr. Bruce Patterson on the call, and we will be presenting some of the presentation together, and he also will be available with me for question and answer. Today's presentation, I will be covering many forward-looking statements, and I'm not going to go to too much detail of Leronlimab. I think everyone knows this story pretty well, but I will say a few things for those who are new, just to understand where we are. Our humanized monoclonal antibody is our lead product, and it has shown potential to have many indications. Starting with HIV, we have combination therapy, monotherapy, and prevention for HIV, which you don't see in this screen. But with combination therapy, this product got fast track designation back in 2006. NIH, National Institute of Health, gave it $26 million worth of grant, which at that time it was used before 2010. We also finished our phase 3 trial and had our primary endpoint, and the FDA met with us and gave us a rolling review. So everybody is expecting to have the BLA, biologic license application, to be filed by the end of this month, and I will update everybody on that in a minute. Monotherapy was the first time that the single agent and the antibody self-injectable at home was tested, and the results got better and better, where FDA agreed to look at a pivotal clinical trial and consider that for a final approval after the first approval, so it will be expanded label. This product also had the indications in triple-negative breast cancer. After we got very strong animal data, FDA also gave us fast-track designation just last year for that. And then we realized that one of the patients who got involved with the compassionate use, which happens to be my own family member from a different cancer, also showed very promising results. Therefore, we filed for a 22 solid tumor cancer, and we got green light for that, which is a very important trial to us. The mechanism of action is the same, and Dr. Patterson will weigh in on that in a minute. But the next trial that we have going, that we just injected the first patient after modifying our protocol, is graft versus host disease. We got orphan drug designation in 2017. So far what I said, any one of these would make a whole company by itself. Any one of these would be very promising for any company. And in regards to NASH, which is non-alcoholic steatohepatitis disease, we show very promising animal study again, just like we did with triple-negative breast cancer, and now we have human data. And with coronavirus, we are very hopeful. Obviously, we have to have data from patients to be able to justify our enthusiasm. Dr. Bruce Patterson will weigh in a lot on that, as he was in touch with the doctor who wanted to use this product for these 2 emergency INDs that we received green light from FDA and got enrolled. Dr. Patterson had to go through the mechanism of action with many people, and the people who talked to FDA had some very serious questions, and he will address those. I'd like for those questions to be posed to Dr. Patterson again on this call, and he will address them. But before that, let's talk about the mechanism of action. It's completely a different mechanism of action for this product for HIV than anything beside HIV, which is immunology's indication and cancer. With regard to HIV, we block the CCR5 on a T cell. HIV that can't get to the cell can't copy itself. That's why we have such a fantastic results in all of our studies. In regards to cancer, it's a 4 different mechanism of action, which Dr. Patterson will get to that in a minute. But our HIV, before anything, the combination therapy hit our primary endpoint, and we are very excited to hopefully submit our final 2 portions of our BLA, which is still being put together. Hopefully by the end of the month, we will be filing that. Then the question comes, okay, you got what everybody wants to do in a biotech world. You got a product to where you had your primary endpoint. Now how are you going to launch the product, and how are you going to come up with the money to manufacture that? Samsung Biologics agreed after reviewing our data that we have a strong situation, and they deferred the payments, which allowed this little company, CytoDyn, to go forward and be able to manufacture a very large batches of product and have a deferred payment, most of it post-approval, hopefully. And then launching a product will cost $50 or $70 million. We were fortunate to have a licensing agreement with $87 million upfront, milestone payments, $87 million milestone payments, and $4 million equity investment at the signing, which we received, and some other initial payments that we received already. So that company will spend all the money that needs to launch the product. Obviously, if we did it ourselves, Syneos Health reports showed that we needed $50 to $70 million to launch the product correctly. That company will do that, and we will receive 50% of the net sales plus cost of goods plus 10%. So now let's go back to the what we have. In regards to combination therapy BLA, there is a lot of question. Why do we keep saying that we probably going to get this filing done by the end of certain months, and it keeps dragging? And as I have told everybody before, we will update you with everything that's happening and give you our best guess based upon what we have right now. That's what you, the shareholders, commanded me to do. You instructed me to give you guys information, and I do. So when we had our BLA submission talk back in 2019, we were hopeful to have it done September of last year. But then when the success with the higher dose came, the FDA was kind enough. That was a big favor for us, that they allow us to submit the same BLA, but for a higher dose, and they require to have 24-week data of the higher dose from another trial, which was very easy to enroll. We could not enroll with combination. It would take us 5 more years to do that. So they said, give us 100 patients from 700 milligram that everyone have gone 6 months. So that delays you about 6 months, and also the amount of time that takes to enroll those patients. We did it in a spectacular timing according to everybody who's involved. And if nobody believes that, I understand. People want it to happen so quickly, but these are obstacles that we have to overcome because of the success of this product, not because of failure of this product, because of the amount of success we got. We have single agent therapy, monotherapy that showed powerful success. So With that, we finished the 100 patient, submitted to the FDA. Now FDA has to find out how they're going to process it. There's two different clinical studies. Usually it's one, but it's now two with two different criteria. So we had to go through a lot of going back and forth to figure out how we can be in the same page as the FDA. Finally, we received the FDA blessing to what they need exactly and how to put it, which format to put it. We received all of that just about 10 days ago or so. And our clinical department is very confident that even though it's a lot of work, we are able to submit it by the end of the month. Then we go to the manufacturing. We had to go meet in manufacturing facility in Seattle. Dr. Nitya Ray, who has done a fantastic job. I can never thank him enough. And the shareholders will see at the later on time when we go through the whole detail of what happened, how he managed everything so well after everything was going down the wrong direction. So with that, they agreed, AGC agreed to give us all the information that we need as soon as they can. Again, that manufacturing got delayed because the vials before were 350 milligram, lower dose, 1.4 milliliter, but now the dose was 700 milligram, which required 2.4 milliliter vials. So you have to have a 6 months of stability. You got to make those vials, you got to manufacture them in that way, and then put them on the stability for 6 months. FDA agreed to receive 3 months first, and then the next 3 months during the review. To make the long story short, we will feel very comfortable to have that done by the end of March. So with all of that, we feel very fortunate to be in a position to file that by the end of the month. Anything that deviates us from that, I will be reporting to you, the shareholders. So in regards to our triple negative breast cancer, we have had very strong results, and so far our number of patients that we have received are 3 patients in our phase 2 have been enrolled, 7 patients on compassionate use, and 2 patients in emergency IND, and that's 12. And there's supposed to be 2 more patients coming in, which will make the total 14. But so far we have 12 patients. That's plenty of data to be able to look at the progress of the patients and be able to talk about it. But I'm going to read you 2 testimony before I go to Dr. Patterson to explain the mechanism of action here. But one of the patients gave us a testimony that I'm allowed to read to you. The testimony said, "So far my experience with Leronlimab has been very positive. I didn't expect to be so easy and tolerable with virtually 0 side effects. The results so far have been super impressive. I'm very grateful to be part of this clinical trial study, and it really makes me feel hopeful that this otherwise fatal disease can be turned into a manageable disease in the near future." The next patient said, "She comes in today with this is the doctor saying, I'm sorry. She comes in today with an astonishing reduction in her chest wall disease with only faint erythema on her chest now, dramatically different than it was when first started." The patient that is my own mother-in-law, as I have said before, that patient was not given more than few months to survive. The radiation that she received back in June ended up being positive, and she was able to make it to November, when then she received Leronlimab. And her CTC after 10 weeks of Leronlimab was 0, EMT was 0, and she was feeling very strong, and she's doing very well so far. But with that, I'm going to ask Dr. Patterson. Dr. Patterson, could you please be kind enough and explain why are we having these kind of results with our cancer patients, and what is your take on these results? I'm sorry, could you start again? Go ahead.
In the cancer patients, what we're seeing is CCR5 positive infiltrates of T cells and macrophages, these specialized cells that are part of the immune response against cancer. What happens over time is that, you know, when the immune response is brought back by other drugs or on its own, the immune response is inhibited eventually by T regulatory cells. These cells express CCR5 and are blocked by Leronlimab and kept from coming into the tumor and inhibiting the immune response. In addition, Leronlimab repolarizes macrophages, a specialized cell type of the immune system, and changes them from a type that is helping the tumor proliferate to a type that has anti-tumor activities. So there's two modes of action of Leronlimab against cancer.
Perfect. And I want to emphasize one thing that we owe Dr. Patterson, great amount, because what happened was at the beginning of this trial, triple negative breast cancer, if everybody remembers, we were not enrolling for close to a year. And Dr. Patterson had argued way before a year was up at the very early stage that you don't look at the tumors having CCR5 only. You also look at the macrophages around the tumor and the leukocyte infiltration. If they have CCR5, enroll them. And his take was that it will potentially have a great result if you do, because they're packed with CCR5. After about a year, finally, we decided to do that. And we trusted his judgment, and the laboratories, all the patients that came in, hardly anybody, maybe one or two, had CCR5 on the tumor cell, maybe a few more, but most of the people, especially the first 6 or 7, had zero. We would have had almost zero patients for a while, and maybe by now we would have had 1 or 2 if we did not listen to Dr. Patterson. And his prediction was solid. So as you can imagine, we are very proud that we were able to turn around a dead end to something that's so spectacular, in my opinion, right now. And that's why I trust Dr. Patterson with our direction going forward with coronavirus situation. So in regards to coronavirus, I have to let everybody know that we announced this morning that we injected the first 2 patients on emergency IND based upon the doctor at the New York, one of the New York hospitals, telling us that they want to enroll them and they wanted to go over the mechanism of action with Dr. Patterson. So he and his whole team had a long conversation with Dr. Patterson. Dr. Patterson shared published data from himself and other colleagues and other people in the world and showed to them why he believes this is very positive. So one of the questions that was brought up between the FDA and this doctor that Dr. Patterson answered was, there are literature out there that perhaps CCR5 antagonist is not going to be working. And Dr. Patterson answered that question where I was very happy to be able to hear that. Dr. Patterson, can you please explain that situation again, why you think CCR5 is important in coronavirus, and the people you were convincing were looking at a different situation? Go ahead.
Yeah, thank you, Nader. The thoughts that were brought up from mice studies with CCR5 completely deleted from these mice, when in fact what we're doing is blocking some of the activities of these cells and CCR5, but not all of them. And I think that's the key point to differentiate between what was cited and what evidence that we have is going to be promising for coronavirus. The fact is, in early to moderate disease, we want to prevent the influx of macrophages and the production of cytokines like IL-6 and TNF-alpha that are causing the destruction of the lungs. And in later stage, in severe patients who were part of this emergency use authorization that we reported on this morning, what we hope to achieve is to break this vicious cycle of cells coming into the lung, producing these toxic chemicals. Those toxic chemicals bringing in more cells to the lung, which produce more toxic cytokines. So we want to break that vicious cycle that's contributing to the lung damage, latent disease, and then in the mild to moderate, prevent that from even happening, and letting the normal immune response take hold, the innate immune response and the cellular immune response against the virus. And all of that benefits also by leronlimab's inhibition of these inhibitory Treg cells too, which inhibit the innate immune response and the cellular immune response. So we're very excited about the possibilities and hopeful for these patients who are being treated, because I think it's these patients in particular, nothing else is working on them. So we're very excited to be able to offer them a possible treatment for their severe disease.
Thank you, Dr. Patterson. So with that, I will not take any more time on the presentation. I would like to go to question and answer. Jack, would you like to go ahead, go over the questions that are already there? How would you like to conduct that?
Yes, Nader, can you hear me?
Yes, I hear you great.
Okay, perfect. Okay, so we're going to start off with the questions that came in from people who registered a little bit earlier, and then we're going to go to the live questions. So anybody, if you have a live question, there's a little function where you can raise your hand, so to speak, and we'll go to you, and you can actually use your mic and audio if it's working to ask Dr. Pourhassan your question directly. So let's go to the first question. Peter is asking, how did the events unfold into having Leronlimab tested in 2 COVID-19 patients in New York? And is there any prognosis?
So for a person who is a former Gilead person, part of Gilead in the past, reached out to us and said that he really believes that this product could do amazing because he knew the science and he believed on all the Dr. Patterson's explanations. And he reached out to his friends and he was very close friend of the person in the hospital in New York, and he's also friends with a lot more, and we're getting a lot of questions now from other hospitals that they want to move forward. And as soon as we heard that, we immediately provided for them anything they asked for. Dr. Patterson did a spectacular job of explaining constantly to all the doctors and the researchers. And then it came down to calling the FDA. So the physician called the FDA and got the IND number, and we immediately overnight the product. They follow all the protocols that are necessary to follow, and we are here now with injection of the first 2 patients.
Okay. Next question is from Seth. I'm trying to read the question. So when, I guess he's asking, when were the emergency CV-19 patients injected? How many injected? What clinical sites recruiting cancer trials? And when will clinical trials get updated? So several questions.
So there is no clinical trial phase 2 on coronavirus starting yet. That trial hopefully will be starting very soon. We hope for the end of next week, perhaps. But the patients that were injected were under emergency IND. These 2 patients are in pretty bad shape. They have coronavirus late stage, and they're in ICU, and they are using Leronlimab. But the protocol that we have is being worked on very carefully. Sites are coming to us right now. We have our own site that Dr. Jacob Lalezari, who's our interim chief medical officer right now, has set up the room for the patients to come in, and we haven't got the green light from the FDA yet. As soon as we do, we will initiate that. But those are different patients. Those are early or moderate patients, not late. There's 3 stages. So stage 1 and 2 are the patients who will be involved in that randomized 2-to-1 placebo 1 product 2.
Okay. Next question from Raymond, asking, what is the status of the BLA and the BTD in cancer? These milestones should have an answer by now.
By the end of this month, we are very hopeful, and we believe we have gone through all the, we have gone over all the concern, the areas of concern that we needed to address, and all the data that we needed to have. The area of concern was to get the data. There wasn't like any concern beside that. And we got all of those together, ready to go forward. Breakthrough designation, as we announced, we don't have enough data. There was one patient in metastasis triple-negative breast cancer, and we need to get more patients, and the FDA wants to have us request a preliminary meeting with the FDA. We will be doing that. Obviously, with the coronavirus situation, we have 12 patients injected, so we want to get data from all of them. The last patient who got involved, we need to get 2 week or 3 weeks data, and then go to the talk with the FDA, and we might ask for breakthrough designation at that time for solid tumor cancer for the data that we have, because we hopefully are injecting the first patient in basket trial soon. But again, with coronavirus situation, we might have delayed an enrollment, but we are going to go as fast as we can.
Okay, next question was from Donna. She's asking, what are your plans to make this important work known in the media, such as Fox News, CNN, major networks? I guess
so. I believe we have to have results. We are a small company with a lot of people doubting that we have anything for HIV. When we proved that, they doubted that we have anything for monotherapy. We proved that to be wrong. And then cancer, nobody believed in it. They thought we're just saying cancer because we want to go in a field that is very popular for biotech companies to be in. We showed that the results, the data speaks for itself, and now we are going forward with everything else, and we will let everybody know as we know everything.
Okay. Okay. So Ben. Okay. Ben was asking, is it being tested currently in Italy? If injected in the NYC in only 2 very serious patients, does that small sample pose risk?
So there's always a risk when you do a clinical trial, but Dr. Roscani from Italy has already talked to us, and they're working through all the regulatory with the EU regulatory right now as quickly as they can. We also have people from England telling us that there are some special condition where we can just ship the product to the doctors. We have to verify that, check with our own FDA in the United States, make sure we are aligned with everything. But we're working on all of those aspects as soon as we can. And the results of these 2 patients, when Dr. Patterson analyzes everything, then we'll know. Dr. Patterson, you are analyzing samples. Could you explain that to everybody, please?
Yeah, so we are very excited this morning to come in and receive patient samples from the 2 patients that have been injected. What we've developed for CytoDyn at IncellDx is a suite of companion diagnostic tests that really show the efficacy and safety of Leronlimab. And in addition, as it relates to coronavirus, we're also able to show the immunologic profile and how that changes on Leronlimab from one that is damaging to lungs, that is to a immunologic profile that is more consistent with an effective immune response. So all of our labs at IncellDx are busy working on those samples actually as we speak. We've scaled up and we're ready to receive additional samples. But it begs the question, again, I mean, we're all talking about tests to say yes or no if you have coronavirus. But the fact is, we have to think about this not in series, but in parallel. We also need tests to show if people have recovered from coronavirus and thus have partial immunity to reinfection to deploy those people in the workforce. But also, we also have to keep in mind that to develop diagnostic tests that actually show that these complex immunologic therapies work is also paramount in these times. So this isn't just about do you have coronavirus or don't you? We need a full suite of assays to be able to show that we're actually being effective in how we treat these patients.
Thank you, Dr. Patterson. Next question, please.
Okay. Okay, so we got Ben asking, what's the status on the HIV BLA, and are you aware of this week's news about a long-acting HIV injectable?
No, No, I am not. The last news from long-acting injectable came that they had a problem with the manufacturing, and therefore their application for approval was denied by FDA. I don't know if there is any update to that.
Okay, I'm going to actually go to, we had a question that was emailed. Okay, I'll go to that later. Okay, so let's go to the live audience right now. Okay. Okay, just 1 second. Okay, I'm just clicking through the questions. A lot of them actually were same as we asked before. Okay, let me go to Tom. Tom, if you are there, let me go to your question. We'll go to you live. Okay, sorry about it's taking a while to scroll through here. We got a lot of people on this call here. Okay. Tom, okay. Okay. Tom, are you there?
I am here.
Okay, ask.
Can you hear me?
Dr. Yeah, we hear you.
Yeah. So, do all tumors have this CCR5, or is that a requirement in order for it to work?
Dr. Patterson, go ahead, please. Yeah, that's actually a great question, because that's what was, you know, we were originally locked into only looking at tumors that express CCR5, and as Dr. Corazon mentioned, we've seen very few of those in the probably close to 50 patients that we've screened. What we're really looking for are these immune infiltrates of T cells and macrophages that are fighting off the tumor that express CCR5 in what's called the tumor microenvironment. So it's not so much whether the tumor is expressing CCR5, but are we getting an immune response with cells that are expressing CCR5, and those are the ones that seem to be responding well. And no, we haven't seen these infiltrates in all tumors, but I would say over 90% of them do have these infiltrates.
Great, thank you.
So that means, okay. So that means that there might be cases where it wouldn't work then.
Yeah, like
is that correct?
I think I've seen two cases so far that don't have these infiltrates. It doesn't mean that, you know, they're not going to get those infiltrates depending on the stage of the tumor. So, you know, it's a situation where we could keep monitoring the tumors, and once they have infiltrates that that presumably are responsive against the tumor, and there are these T regulatory cells that are trying to inhibit that, that would be certainly a time to initiate therapy. But there's nothing that says that we can't continue to monitor those patients to see if they do develop these infiltrates.
Okay. Let's go to the next question. Okay, so it's so just please be patient. We got, I think we have about 30 questions here. We got about 500 people on the line. So these are the questions. Okay, so we'll try to go quickly through these. Tom was asking, a long-time shareholder here since 2013. Thank you so much for your continued effort to keep investors informed. These updates are quite reassuring. Okay. Okay. Adam is asking, do you have any sales projections for 2020? And please explain 50% revenue sharing.
So we don't have any projection we do have projection, but we don't want to share that this time till we are 100% sure what we're doing exactly, because the cancer situation could get expedited. Coronavirus situation, we have 2 offers right now from China and Taiwan that they want to buy every vial that we have. and we are not accepting that. We didn't even continue when they said that, because we believe that there is a lot of potential here in the United States. And in regards to the sales for the 2020, so we will just update everybody as we find out exactly which one we're going to go forward with. In regards to the amount of, the 50% that we're going to get, the sales, when that happens through the licensing agreement that we have, once that sale is made, 24% or something comes off of that price for Medicaid, and then 9% about rebates. So about 33 to 35% comes off, and then we split that to half, and the cost of goods, goes in and out, so we get 10% more. There's a formula for that, and we also receive our milestones. So the sales happening as they're happening, we get milestones.
Okay, Stephen was asking, what's the status of overseas licensing in China, Taiwan, South Korea, Japan?
We have slowed down China and Taiwan's going forward, because we now are really focusing on the coronavirus situation, because of the mechanism of action that we believe is very strong. We are going to see what happens in the United States. We like to use every one of those vials if the results are positive here in the United States first. We are going to get 100,000 more vials from Samsung within the next 4 to 5 weeks or so, or we are expediting receiving those vials, which will put us in a different position. Then we will continue. But the talks with Japan is still going on. Talk with Taiwan is still going on, and China the same thing. But we are just pausing from our side.
Okay, Gary's asking, what's the difference in effect of the Regeneron remdesivir drug for coronavirus and Leronlimab? Does the Regeneron drug obviate the need for Leronlimab?
Dr. Patterson, could you talk about the Regeneron drug versus Leronlimab? I think they're antiviral, I'm not sure.
Yeah, and I think they're just two completely different mechanisms of action. I don't know that there's another drug out in the market. Some people are saying arthritis drugs which target IL-6, but IL-6 is just one of many cytokines that are being made in the lung, causing damage. The other big one being tumor necrosis factor. And so I think the great thing about Leronlimab is it inhibits these pro-inflammatory cytokines, but it also enhances the innate immune responses. So it's got a multitude of immune modulatory effects that are very positive for the disease process, and I think that's what we're most excited about.
Okay.
Thank you.
Okay, so Tony's asking, how are the two virus patients? Have the two virus patients been diagnosed with ARDS prior to the injection, or has the disease not progressed that far yet?
Dr. Patterson?
Yeah, so these patients have severe lung disease, but it's not frank ARDS yet. And I think, again, what we're trying to do is break up this vicious cycle of immune cells coming in, producing cytokines, which bring in more immune cells, which produce more cytokines, and break that vicious cycle before they get irreversible lung damage. And so we're very hopeful about that possibility.
Okay, so Samir is asking, he's saying, I think Dr. Patterson touched on this, but in regards to the coronavirus, what is the plan to enroll mild to moderate patients who may be more likely to respond?
So we have our own site, as we said, Dr. Jacob Lalazari, who's working very hard to get that site ready to go. We don't have a green light from FDA to start phase 2 coronavirus. We hope to get that next week, we were told by Dr. J. Lalazari. And once we get that, we do have requests from quite a bit of different sites to enroll, and we're working to have those in parallel. So we'll let everybody know hopefully next week.
Okay, Henry's saying, not a question, but I just wanted to say how much we appreciate your hard work, Dr. Pourhassan and Dr. Patterson.
Thank you so much. We appreciate it.
We got a bunch of same type of things, a lot of comments like that, so positive comments. Dr. Porras, we got about I think we're going to have, like, I'm scrolling down, and we got about 40 questions. I don't know if we have time because we have another presenter. Do you want to take a few more, and then we'll maybe we'll actually address all of these probably in a follow-up interview where you can go through them. We have like a ton of questions.
Well, let's have just 2 more, please. Okay. We got a very busy day, and Dr. Patterson is in the mix of testing a lot of important samples for us right now with coronavirus. So, but go ahead, please.
Okay. Okay. So Randall's asking, will you update us on the financing?
Yes, we are hoping to announce something about that. We are working with a deal that we really think is a great deal for us. I think every shareholder would be very pleased with it, but we just can't talk about it because we're in the mix of negotiation right now. But as the shareholders know, we have been blessed to not fail with funding at any time in the last many years that we've been in the country.
Okay. Kelly's asking, I think we've got a couple of questions very similar here. Which is, when will you be uplisting?
The uplisting, as we have said before, New York Stock Exchange and NASDAQ. If I was going to do that right now, if we were going to do that right now, we would be needing to to raise money to last 18 months for New York, 12 months for NASDAQ. New York, we talked to the director of the uplisting, Paul Dorfman, and he said that they're not even going to give us any credit for the sales that we're going to have. So they're going to assume that we're not going to have any sales, which is absolutely, to me, very, very difficult to work with. So I'm giving up in New York Stock Exchange, probably just work on NASDAQ. But the price point for New York is $2, NASDAQ is $4. I don't think shareholders would like it if I say to everybody, we're going to raise $100 million at these levels. I always was hoping, our team always was hoping that we get to a better position for better raise. We have done that and got out of the 30 cents level. Now we're in this level. We think the future is very, very bright. So we're not rushing to jumping and raising a whole bunch to uplist. I think the science is so powerful, in my opinion, that we're not going to have any problem raising money at a higher level. And that's what I'm hoping for.
Okay, Dr. Porras, thank you. And we'll be doing a follow-up with you probably next week, and we'll go through all the questions. We must have, I think, it looks like 50 plus questions, so we'll get a lot of people here. Again, Dr. Porras, thank you. And everybody, let's meet our next presenter.
Thank you so much. Thank you, Dr. Patterson.
Thank you. Thank you, appreciate it.